Hello everybody, I am Patrizia Fattori. I am a professor of physiology in the University of Bologna in Italy. And as you see from the name of my department, I have a neuroscientific background. And today I will tell you something about our studies in the grasping and manipulation domain. And I think this can be a very intriguing topic because our hands are centrally involved in many of our daily activities, and reaching for objects and manipulating them is an almost effortless activity, but it is very, very simple. So it can also be accomplished by a little kid. So it's on one side a simple task, on the other side is very, very difficult because the sophisticated function of the human hand relies on more than 20 independent degrees of freedom, actuated by more than 30 muscles and aided by numerous proprioceptive and tactile sensors. And this has remained for a long time beyond the reach of any human knowledge. So the complexity of this task is probably the reason why this ability has been associated with divine abilities, like in this painting here. But don't worry, today I will limit my talk to grasping and manipulation in humans, not in God. When we plan to open a bottle, we anticipate within our brain that we will assess its content with a very familiar sequence of additional actions, for example, involving eventually other objects such as cups or spoons. And not only can we anticipate these actions, but we can imagine that what we will feel when the bottle will be in our hands, and any deviation from our expectations triggers a rich repertoire of corrective actions so that we can finish most of our daily activities very safely, despite the absence of really precise information about physics of the world. So we are not aware of the processes going on, but we take into account many, many variables. For example, to grasp a ripe tomato, visual information on the tomato size, its shape, its orientation is used to determine stable placement of the digits. This makes us grasp the tomato with digit placement so that to intersect the object's center of mass, ensuring that the forces of the digits are applied to the center of mass of the object. The eyes are directed to the location where the index finger will eventually land. So we anticipate with the eyes the location of the landing of the index finger. Choice of grasp contacts requires the integration of anticipatory control of digit placements with digit position dependent force modulation. And the grasp is shaped not only by the visual appearance of the object, but also by what we plan to do with the object subsequently. So for actions that belong to sequence of actions, we have in mind the following step involving our hands, so to optimize the end state comfort. So for example, in this case, if we see a glass full of wine, maybe we would like to drink it and we grasp in a certain way. But if the glass is empty and dirty, we can grasp it with an awkward reverse orientation of the hand for the starting grasp of the object, because we have in mind to release it in the dishwasher, and we anticipate the comfortable position we will have when we will reverse the glass into the dishwasher. So to finish the action with the hand in the comfortable normal state. So this indicates that our grasp choices are informed by rich background knowledge about the object and non-trivial understanding of the purpose of the grasp. But where do these processes occur in our brain? I must say that most of our knowledge about brain control of hands stems from studies with monkeys, such as macaques, as here in the picture, which share with us the ability of fine manipulation. A vast body of studies on macaques has led to the discovery of a complex network of brain areas involved in the recognition, preparation, execution, and learning of manual actions that is now well known in macaques, with additional findings in humans that point to the existence of homolog networks in the human brain. So let's go to the brain. Many sites are involved in these complex processes. One of them is the posterior parietal cortex that we see here in color for the human brain, and it is the part of the brain that is located here, and here with the colors for different areas in the macaque brain. First, I must say that the two pictures of the brains are not in the same scale because, of course, the macaque brain is much smaller. But what you see is that we have a huge part of the cortex that is organized in parallel ways. You can see here that you have a higher number of areas, more areas in the macaque than in the human brain, not because they are differently organized, but because we have a better knowledge of this cortical mantle, and we are still using this knowledge to better discover the areas of the posterior parietal cortex in the human brain. But we will not focus on the number of names or names of the areas. I want to drag your attention on the fact that this part of the brain, the posterior parietal cortex, is thought to be an important nexus in motor planning and online control of prehension, so of grasping and manipulation, because of its anatomical connections with both the dorsal stream areas of the visual system, but also the anteriorly located somatosensory areas of the anterior parietal lobe. So this part of the brain can combine visual information and proprioceptive and tactile information to coordinate grasping and manipulation. And this has been known since the early studies from a neurophysiologist, a great neuroscientist, Vernon Moncastle, that in the 70s established that neurons in posterior parietal cortex play a significant role in reaching, pointing, grasping, and manipulation. Grasping is a unitary action composed by different stages, different aspects that complexly lead to prehension. If we see in this kinematic recording that we do in our lab, but everywhere in the world you can see this, that while for reaching and manipulation, first you go with the hand in the location of the object to be grasped, and then you shape your hand according to the shape and orientation of the object. And traditionally, the two processes, the one of reaching and the one of grasping, which are two components of prehension, have been considered separately, have been studied separately, and this brought to the model summarized in this picture. First, I let you notice that this drawing is taken from the Candle Neuroscience book, which is the Bible, the textbook that every PhD student in neuroscience needs to know. So it's our textbook for neuroscience. And in this, the last version of the Candle, we have a connectional scheme, so a circuit in between the dorsal part of parietal cortex and dorsal part of premotor cortex that is considered to be a circuit for reaching, and these are the names of some of the areas involved, and another circuit more lateral linking the inferior parietal cortex and the ventral premotor cortex, that is a circuit involved in grasping. So this classical view describes two separate channels for reaching and grasping. But the situation is more complex because I will show you that grasping neurons have been found in dorsal aspects of parietal cortex and also of premotor cortex. So the two circuits are not segregated, are unified. And I let you notice that the well-known FARS model by Arbib and colleagues deals only with this lateral part of the circuit, ignores the first part, because at that time it was not known the things that I'm going to tell you now. So what we did in our lab was to test in the same parietal area the entire prehension process, studying the elements that drive our hands toward the spatial location of the object and the parameters that conform our fingers to the object to be grasped. For example, by starting with the first component of the prehension, which is directing the hand to the location of the object, so the reaching component, we have found neurons, like the example shown here, whose discharge is tuning according to the direction where the hand is going. In this case, the activity has been aligned on the onset of the reaching action, and we see a very nice peak of activation when the action is performed, and this peak is tailored to the direction of the reaching action. This is the discharge of just one cell. Of course, we recorded hundreds of neurons, and we found each neuron with its particular spatial preference. But anyway, the discharges in every case, were tuned according to the location of the target, so location of the hand. Then we went, as I told you, a step further, and we have tested when you reach and grasp an object with different orientations in the same spatial location. So you don't have spatial parameters. You have to consider the characteristic of the object, its orientation. Is the neuronal firing in this parietal area tuned by this parameter, grasp orientation? This neuron here shows that this is the case because its peak of activation, aligned again on the onset of the reach to grasp action, is sharply tuned to the orientation of the wrist to grasp in the same way, in the same spatial location, a certain object, but with different wrist orientations. So this additional element of grasping is encoded in this parietal side. We went a step further and we checked whether, without changing wrist orientation, but changing the shape of the fingers that grasp objects of different shapes, it is contributed to the modulation of these cells. Here I show not a single neuron, but the population discharge. And this is the onset of the reach to grasp action. And you see that this discharge is very well tuned in the entire population according to the different grips performed. And here I show you a very rudimentary grip, a whole hand prehension, when you grasp with all the fingers the entire objects. An intermediate grasp in which you use a part of the object, a handle, to grasp it and to use it as a tool, or a very fine and precise grip in which you just use the first two fingers to conform to the shape of the object. And the population is able to distinguish between the different grips. I let you also observe that in this part, well before the action execution, you see this is 1 second before the onset of the movement, the discharge of the neurons start to diverge. So it means that during the planning of the action, there are parameters that are took into consideration by these cells, that by this area. So at the end, we can say that in a unique parietal area, we found the neural machinery able to perform the entire prehension from reaching to grasping. And this can be considered the neuronal counterpart of the kinematic observation made by another great neuroscientist, Marc Jeanneret. About 30 years ago, he found that dexterous grasping, hand-arm kinematics, is highly stereotypical, and it has a pattern of gradually increasing of grip aperture to a maximum value that is highly correlated to the size of the object to be grasped. And this maximum grip aperture is reached near the last third of the movement, after which it shrinks until the fingers make contact with the object. And this pattern has been confirmed in many subsequent studies. So the two processes, reaching and grasping, are interlaced, and what we have found in our parietal area is that the processes can be encoded, all these processes can be encoded in a unique parietal hub, but of course it is very useful to have more than one hub in the brain that encode this information and where the entire prehension is coordinated. To these three parameters of grasping, we can add another layer of complexity, considering also the presence or absence of visual information for grasping. So usually our visual system extracts the visual information necessary to plan and orchestrate grasping. And we studied how visual information and grasp information are wired together by studying, for example, how the same object with different orientations is grasped in the light or in the dark. And we checked whether neuronal firing was considering these parameters, and we used a data reduction technique, DPCA, that takes into account the contribution of single parameters and their interaction in the population activity. So we see first that DPCA has a similar ability with respect to PCA in our conditions to explain the variance of the discharge of our cells. And in addition, we see that it is able to capture the weight of visual condition, wrist orientation, and the interaction between these two conditions, showing that wrist orientation and visual condition have a similar weight in explaining the variance of our neuronal population. We did the same also in a more complex context in which the action has to be performed toward 5 different shapes of objects requiring 5 different grips, from the most rudimentary, the whole hand prehension, to intermediate, in which you have to insert 1 or more fingers into holes of the object in order to grasp it, and till the most extreme one, which is the grasping of the cylinder into the groove. So you have to orient the first 2 fingers so that to align to the groove and to insert the first phalanx of the index and thumb in order to finally grasp the object. So again, with DPCA, we found that we can explain the majority of the variance of the population, and visual condition, grip type, and condition interaction had almost an equal weight to explain population variance discharge. So it means that the neuronal firing is able to capture also the richness of visual and sensorimotor information that are linked together to control the action of grasping, but also to advance, to anticipate the outcome of the actions in terms of haptic sensibility, proprioceptive information, and the visual checking of what is going on and if it is correct with respect to the expected outcome of the action. Of course, when you have vision available, you have all this information at your disposal. When you are in the darkness, you have only the sensorimotor information, not the visual one. And with this task, we went on also to perform a decoding task in which we used the neural discharge that we observed for the 5 different objects. Maybe here it's not very clear, but these are the objects. So the discharge to grasp these objects, the observed discharge against the predicted one, in the dark on the top or in the light in the bottom, in different time epochs of the task, when the monkey was simply observing the object before performing the action, when in the delay, when it was preparing the grasping and during grasping execution. And what we found was that in all these conditions, our decoding ability was really very high, reaching a maximum in the delay or in the grasping execution, also in the dark, but practically 100% in the light. So it means that indeed this neuronal finding is very well able to inform us about the kind of grip that is performed. And all these discharges have been found in the most dorsal part of parietal cortex. So in the part of the parietal cortex where, according to Kandel, only reaching was expected to be present. Indeed, you see that these parameters have nothing to do with the reaching because the position of the object was always the same. So there were no special parameters involved in this task. Okay, of course, we can add other layers of complexity. For example, attention. In this case, we can see that grasping the act, very simple act of grasping an apple, can begin with the shift of our visual attention and usually also the shift of our eyes to the direction of the object. And so our visual attention, our retina also drives and prepares a coordinated the action of motor commands to the hand in order to reach and grasp the object. And this action comes with the richness of sensory motor and visual feedback that we have spoken about earlier, but also more complex sensory motor feedbacks, like for example, a touch on another part of the body that has nothing to do with the apple or with the action we are performing. But attention is another story. And you need to invite me for another meeting so I can tell you also this part of the story, because these parameters also is encoding in this parietal side. Anyway, the thing I wanted to convey to you is that when we perform a very simple action like the one of grasping an apple, that sometimes, like in this case, can be very dangerous, but all the complexity of this action is hidden from our conscious perception, making us entirely unaware that our brain has just performed a highly amazing act. And an ultimate understanding of how this became possible might be sought in the underlying neuronal processes scattered across the brain areas that are involved in this action. And fortunately, in the last decades, single-cell studies have been combined with modern imaging methods and revealed a substantial number of brain areas involved in these processes. Finding homologies, for example, just to give you an example, the area I've spoken you so far is this area in the macaque brain, area B6A, and in collaboration with Sabrina Pizzalis in Rome, we have found the human homologue of this area in our brain. And I don't want to go into the detail of the location of this area, but it has been shown also in relation to the other areas of the dorsal visual stream, its location, this you see in the 3D reconstruction of the brain, the location of this area, and the functional properties that we can test in the scanner so far indicate that this area is exactly with the same physiology, with the same neuroscience as the macaque brain area. Of course, our interest is not to know the macaque brain, but the physiology of the human brain. This is why, with different techniques, we investigate this in these two primate species. Other laboratories, in addition to ours, has found this parietal side involved in tasks dealing with reaching, like in these two works, but also dealing with grasping or the part of grasping connected to wrist orientation. So there is really an homology between this parietal side in the human and the macaque brain, and this is very important for us to improve our knowledge of how our brain organizes grasping and manipulation of object. So I hope that with this talk I have had a more human approach to the grasping action, and it is less divine after hearing my talk. And before leaving you, I want to thank all the collaborators that along the decades have contributed to these results, because you can imagine it is a long work to perform. And I can take a question that I received, that is this. The main reason why humans outperform robots in manipulation tasks are two. Because of their capacity to plan motor actions, or because humans can rely on thousands of sensors. Since robots are still quite different to humans from the hardware point of view, is it worth to exploit neuroscientific studies to design skilled robots? So, as I was saying at the beginning, the human hand has 27 degrees of freedom and articulates 27 bones with 35 muscles. So it's a model of dimensionality reduction, both in terms of movement synergies and in terms of optimal control signals. The point is how the limited neuronal resources that we have can control a very complex system with so many degrees of freedom and reaching so many, so a high efficacious control of it. I think it's not a matter of hardware and of sensors, but of organizational principles. So the brain, how it is made, how it is engineered. And so far, we have only a limited knowledge of the processes going on in our brains to smoothly control grasping and manipulation. And this is why neuroscientific research can be very informative for many disciplines, one of which is robotics. This is why I think neuroscience is really a wonderful research field that can have many outcomes, many consequences also in very technological disciplines like yours. So although I can imagine that even non-humanoid solutions can be achieved for dexterous manipulation, reproducing the human dexterity in robots can be very useful, at least in the fields in which you have to have human-robot interactions that I think will be every day more important in the next future. As a neuroscientist, I think that if a robot behaves like me, it is easier for me to interact with it in my daily life. This interaction will be safer, more immediate. So the short answer to the question is, I believe it can be useful for dexterous robots to be inspired also by human sensors and actuators. But I suggest to concentrate more on the brain circuits dealing with them. But of course, this is just my opinion. And I thank you all for your attention so far. And if you want to contact me in the future, this is my email. I'm very interested in collaborating with you and in having this mutual information to improve grasping and manipulation as humans are able to perform.